the 2017 bucket courses. Delighted to have a room full of people this morning, and I know it's going to be worth coming out in the cold today. I'm Joanne Bungie, and I'm, well, I guess I'm the head bucket. All of the buckets on our planning committee have been busy during this holiday season, preparing for the winter and spring seasons of the bucket courses. You'll see on the slip that you have on your chair this morning what the winter season is, and I know you're going to be enjoying it. I have one announcement to make while you are silencing your cell phones and turning on your tea coil, and that is that this week and next week we will have the regular times for Clark's bucket course. He is, as you probably know, an active faculty member at the college, and so he is going to be teaching early, well, today and the next week. The students are not back on campus, so he is free to come and teach us. The last 2 weeks, January 25th, I believe it is, and February 1st, he will be teaching at the college, and he has an early morning class and an 11 o'clock class. But he is willing to sandwich us in between those two. But we will have to accommodate his time a little bit, which I know you'll all be willing to do. We would like to start the class at 9:30 on those 2 mornings, and then we will close the class at 10:45 so that Clark can get back to the college for his day job. So that will, not next week. Next week will be just like this week, but the last 2 weeks. And we will send you an email to remind you of that as well. So just kind of keep that in mind.
I thought I was going to get a police escort.
You deserve it. We are so happy to have Clark Lindgren back again. And we have responded to your many requests for having him. And we're happy that he is able to be here today. He is, as you know, professor of biology at Grinnell College, Patricia A. Johnson, professor of neuroscience, and I was professor of the year. We are so happy to have someone of his caliber interested in doing the bucket courses, and we're delighted to welcome him back again so that he can tell us about neuroscience in terms of synapses and how they can make and break us. Please welcome back again Professor Clark Lindgren.
Thank you very much. Can everyone hear me? Okay, good. I say this every time, but this is my 4th bucket course. Is that right, Joanne? That sounds like about like that. And I say it every time, and I'll just say it again. I really applaud you for this. This is a great venture, and I guess particularly the organizing committee for pulling this together every year. I think this is a really wonderful thing for a community, and I'm very happy to be part of it. With technology, hopefully we'll get through this. We're going to try a couple things here a little different. So, to start, it's my daughter. Some of you know her, Hannah. She's a freshman at Lawrence University this year, having a great time. The reason I put this up here is to remind you that, or to confess to you that yes, I do have a vanity plate on my car. And it says Synapse, and so why is that? Well, I've been thinking about and studying synapses for 35 years. Where does the time go, right? I've been doing that for a really long time, and telling people, whoever will listen to me, which is usually students and then granting agencies, that the synapse is the most important, essential part of the nervous system for us to understand and to get right. And that's not a crazy idea. That is generally, I think, I've received a warm response to that message. But I've never taught a course that's just on the synapse. So this is the first time, hopefully not the last time, I try to do that. But if I do like how this works out, I might try to develop this into a course that would be something for what we call non-majors or non-science majors. I think it's a nice way to sort of introduce some of the key important ideas in neuroscience. So this is either the first of maybe a few more, or the one only. We'll see. You can decide that. All right. So here's the itinerary for the course. So I'm going to start off telling you what a synapse is, how it looks, how it functions, and I'm going to give you, not just tell you what we currently know or think we know, but also give you some insight into how that was figured out. I think that's really an important thing that makes science different from so many other ways of describing it. I mean, I'm thinking politics right now because that's on my mind, but in science we do have this expectation that we get our ideas from something that's tangible and is experimentally verified. So we're going to talk about the synapse and how we know what we know about it. I'm going to try to tell you why synapses are so special, so what makes them so unique, and hence my enthusiasm that I've maintained for all these years. And then ending up, how do synapses make us, and then how do they break us? And I'll try not to end with a completely negative, I'll try to bring it back to a positive thing at the very end, but that's kind of the general strategy that we'll go through. And I'm envisioning these will be roughly the 4 lectures, but if I get out of sequence, they may not actually line up precisely among the 4 weeks that we have, but that's the plan. All right. So before we get started, preliminaries, what is the self? So that is actually a question. So how would you define self?
Unique being.
So you're a unique being, okay? Any other ideas? What is?
Ways you feel and react.
The ways you feel and react.
Sort of a whole personality.
Personality? Sure.
Complex.
What was that? Complex. You think we're complex?
Yes.
I agree, yes.
The ability to distinguish self from other organisms.
Or even other selves.
Yes.
Right, so have a sort of a sense of
From a
identity.
single cell independent organism to
Okay, so an
being.
identity that's distinct from other selves, or which could be humans, which could be non-humans, whatever. What could you lose and lose yourself? Yeah, sure. That's too easy. Yeah, right. But think about it in terms of these qualities that we've talked about. And one of them you haven't mentioned yet, and that's what would be probably most. You could lose your personality. You could become a different personality, but you might still be a self. You could lose. Well, I guess if you lost your sense of uniqueness, that would be kind of problematic. But
Something about uniqueness and sense of self versus others. I don't know how exactly consciousness fits into that.
so consciousness is a term that
Consciousness, self-awareness.
makes sense. So consciousness, this idea of. Usually philosophers try to think about that in terms of what it's like to be. So what is it like to see blue? We can describe it objectively and say there's a blue jacket. But what's the quality of, what's the experience of experiencing blue? And so there's those sorts of things. But you guys aren't getting what I think is important.
Memory.
Memory. Somebody said memory. Yeah, I think that, and I have had a mother who died of Alzheimer's disease, and I watched her lose herself. I mean, I don't know how else to put it, but she question?
How about satisfaction?
Okay, so motivations and then satisfying those motivations, those are all things. Okay, so you've got the right idea. So we're talking about pretty much everything that makes us who we are. The most concise definition I came across was written by Joseph Ledoux in a book actually called Synaptic Self. And he basically said it's just the essence of who you are. So that's really what we're talking about when we're talking about the self. So it's a pretty significant challenge to now describe how synapses make that happen. So the premise of this course is that your self, how we've defined it, is created by patterns of interconnectivity between cells in your brain. Now you might think that's crazy idea, you might think it's too simplistic, sacrilegious, or or all the above. But let me give you a little bit of background for where that statement comes from, and then hopefully at least you'll understand the idea that I'm going to be describing in this course. So this goes back to the question that's called the mind-body problem. You raise your hand if you've heard that expression. Okay, so it's been around a long time. People have been talking about this for a long time. It's still an area of serious debate, discussion amongst philosophers. Not so much about neuroscientists, and I'll explain why that is, but the mind-body problem, today, the sort of the contemporary view of how the mind and the body relate is that the soul, or spirit, or mind, whatever you want to call that, and there's a lot of synonyms for that, is separate from the body. That those are two separate things. You have a body, and then you have an immaterial soul. Right? I mean, isn't that kind of what we generally sort of think of when we sort of talk about the self? When you said these things self, you were probably thinking of a sort of a disembodied, or sort of a concept that wasn't necessarily tied to your physical brain. You might be surprised to know that idea is fairly recent, at least in terms of human history, the length of human history. That idea has not been one that humans have held for the majority of our history. The idea of having a separate mind and body was not really one that the earliest Greek philosophers talked about. Later on, Plato kind of started describing ideas that sort of expressing this idea that there might be this different non-material aspect of being. In terms of like early Christian teaching, even, you might be surprised, really emphasized the body more than the spirit or the soul. I mean, after all, Lazarus was raised from the dead, and that didn't just mean his spirit came out of the grave, his body walked out. And Jesus, I think, in Christian tradition, it's still important that Jesus rose from the dead, right? And again, it wasn't just an immaterial soul that rose, but it was a body, and that really reflected the underlying concepts of the first century, which was really both Judaic and then also the early Christians sort of picked up that idea that it didn't make any sense to them to think about a non-material soul, spirit, or mind, or whatever. And then I can't help but point out the Egyptians, who kind of took it to the extreme. They not only thought their bodies would survive death, but they packed along stuff to take with them, including wives and slaves and people like that, right? So they really had this idea. They wouldn't, they preserved their body, and that was all because they, in their minds, no pun intended, they really saw the body as being essential. So the modern, sort of the sophisticated description of this mind-body problem was really, can be attributed to René Descartes, who he's the one who made the famous cogito ergo sum statement. I think, or I know, therefore I am. That was basically as much as he could actually say about anything. My best spin on this is a t-shirt I came across that said, I don't know, so maybe I'm not. I don't want to get that t-shirt. So the mind-body problem, so the way Descartes handled this problem and tried to make it a sophisticated, rigorous description of the connection is he described something called substance dualism. So the idea is that there are two different substances that are completely different. So the body is a material substance, res extensa, it's extended in space. So the body is something that is extended out in space, and it's part of the material world. We understand it in the same way we understand a rock. Or actually, Descartes' day, one of the things that influences thinking very much is back in Paris, automatons were really quite popular in art, and there were even, like in the streets, they had these things, these machines that would carry out these very lifelike type of behaviors, and so he really got thinking about that and thought that's really a good metaphor for what the body is. And then, so it's just a machine. It's like these automatons. Think Westworld, if you want, in terms of these robots that are actually acting like humans. The mind, on the other hand, he argued, was completely non-material, as cogitans. Okay, so it was something that was of a very, very different substance, and that was the part of the human that was sacred. That was the part that survived death. And so that, you recognize that that's really pretty close to the contemporary view, popular view about how these two are related, and we probably really attribute that mostly to Descartes. One of the significant consequences of this, which a lot of people don't realize, is that by formulating this clear separation that had been building, so this didn't just come out of nowhere from Descartes, but this idea had been building, it actually allowed people like Descartes to dissect cadavers. And Descartes did that. He was actually, like back then there weren't people called scientists, but he actually was very fascinated with the human body and dissected brains. And of course that was not entirely cool with the church at the time, because the idea of the body was sacred. And that goes way back to the beginning of Christianity, this idea that the body was as sacred as the spirit. Well, Descartes figured out a loophole, that's my somewhat cynical view of it, and said, well, when the person dies, the sacred leaves, and there's now just the body. It's a machine, just like these automatons that we see around Paris. He didn't really ever explain what this substance was, but he did recognize a problem with his philosophy, and that is, if you have these two completely different substances, how do they interact? Right? So the physical body is perceiving things through the senses, and then that has to get transmitted to this other thing out here, the res cogitans, and then it has to, if it's going to make you move, it has to transmit that back to the body. So there has to be some sort of communication between those two. And he didn't explain how that might work, but he did pinpoint where he thought it happened, and that was in the pineal gland. Does anyone know anything about the pineal gland? It's named after pine cone, by the way, because it looks like it's shaped like a pine cone. Why did Descartes pick that?
Because it's in the center of the brain.
I didn't hear that.
It's in the center of the brain.
It's right in the center of the brain, and it's singular. So most of the brain structures are duplicated. There's a left and a right structure, and they more or less mirror each other, although not exactly. It was one of the few structures that there's only one of them in the brain, and so that seemed to be an important feature of this antenna, is it had to basically have equal access to the entire brain and not be restricted to one side or the other. That was really his idea. And here's, if you want to see an actual brain structure, you can see the pineal gland, and it's right smack dab in the middle of the brain. And it's this little tiny pine cone shaped thing. What we know about it now is it's actually an endocrine gland, and it secretes melatonin, which is actually involved in sleep. So it's involved in wake, sleep cycles, and so forth. So people have speculated why he didn't pick the pituitary gland, which is another structure that sits right down kind of below that. It's not in this section. But the pituitary gland is often, it's this little tiny structure that gets ripped off when you take the brain out. So it probably wasn't even there in the specimens he was looking at. So he picked the pineal gland and talked a little bit about how that was the central place where the communication would occur. Now, today, neuroscientists really don't subscribe to this Cartesian dualism. And in fact, Gilbert Ryle, this British philosopher, early 20th century, kind of derogatorily described Descartes' dualism as the ghost in the machine. You had the machine, and then there was this ghost hiding in there, and it was basically pulling the strings and running the show. That idea is not one that is subscribed to by neuroscientists today. The prevailing view that neuroscientists work on, and this course is based around that prevailing view, is what I like to call physicalism. Some people call it materialism, but I think physicalism is a better description of it. And it states that the brain, and nothing but the brain, creates the mind. And it's an entirely physical process that then creates all of these things that we're thinking of as the mind. I actually would modify that a little bit and say the brain and the body. It's really the entire body, because now we know that actually a lot of our thinking and a lot of our feeling, our emotions, our personality are also really critically linked to other things that are happening throughout the body, not just the brain. But the idea is that the brain, the body, and then nothing else but the brain and the body, that's really what creates the mind. Okay. Now you might say, why do neuroscientists take that position? Well, I have a short answer and a long answer to that. Okay. The long answer will take me about 4 weeks. So just sit back and hopefully after 4 weeks, you'll at least understand why that's a view that neuroscientists hold, why it actually informs what we do and how we sort of think about these problems. The short answer is described best by Joe Friday of Dragnet fame. It's a view that just fits the facts. That's what you'll see. I mean, that view is really the one that fits the experimental data we have, the data from injury, all of the things that have been gathered over the last 100 years or so. It really does fit with this idea. Not exclusively, but the cleanest, the simplest explanation is the physicalism idea, which I'll propose. Okay? So what is the brain? So we've talked about the self, we've talked about the underlying premise of the course, that the brain is what makes the mind possible, so what is the brain then? Well, one of the ways of thinking about the brain is when we're talking about the human brain now, it's about 80 to 100 billion neurons. Okay, so that's a lot of neurons. Now, if you read my other bucket course where I talk about glial cells, you know that there's also even more of these other brain cells that often don't get talked about very much. And I'm not going to talk about them that much in this class either. So, but there also are these other cells. We actually have even more than the 100 billion, maybe 500, 600 billion cells that make up this structure we call our brains. Each one of those neurons is estimated to make about 1 to 10,000 connections with other neurons. So that means every neuron is sending out and interacting with 1,000 on average, or 10,000, and each neuron is receiving, on average, 1 to 10,000 connections. So that means you just do the math. Hopefully I got this right, Burl. Seeing a physicist in the front row makes me scared. In a good way. So I come up with 10 to the 15th synaptic connections, or 1,000 trillion synaptic connections, which is probably more than the number of stars in our galaxy. So it's just literally an astronomical number. And the idea that neuroscientists use to talk about this is you have this network that is more complicated than just about anything, although this is a little bit of a parenthetic statement. The internet is starting to approach that, which is really kind of amazing. I mean, in terms of the connectivity that we have out there, the internet is beginning to get close to the sophistication and the complexity of our human brain. It's getting people to speculate about whether the internet might develop a mind of its own, might become conscious in some way. But anyway, that's another subject. So this is really what neuroscientists think about as being responsible for these functions of the brain. And so, not surprisingly, the National Institutes of Health has created what's called the Human Connectome Project. And you remember the Human Genome Project? The millions of dollars that were spent to actually figure out essentially the sequence of DNA in the human genome. There's an analogous project now that's been going on for about 4 or 5 years. It's called the Human Connectome Project, and it has an even more ambitious goal. And this project is designed to figure out that pattern, that unbelievably astronomical connection. To be analogous to an astronomer saying we're going to figure out where every single star is in our galaxy and how it's related to every other star, some crazy thing like that. So it's a really, really bold idea, but it's been started. NIH has already committed $30 million to the project. It started off in a few places. There's a project now from the University of Minnesota that has 1,200 people that are in the project, and by using different kinds of non-invasive brain imaging, they're hoping to actually figure out, or at least sort of approximate, this connectome, or what this network looks like. And why spend $30 million on that? Well, it's not because people are looking for money to spend, but it's because that presumably will give us insights into how the brain works, and then insights into how the brain makes us. Okay. So that's where that comes from. Stay tuned. Nobody is really expecting this to be done anytime soon, maybe 100 years from now, I don't know. Although I should say that a spin-off of the Human Connectome Project is a lot of laboratories are basically doing the analogous thing on non-humans, and so like the entire connectome has been created for a tiny nematode called Synapses elegans, which has like 200 neurons or something like like that. So that's been done, and other, like this, it's being done in, for example, mice and rats as well, which don't have quite as complex a connective. But anyway, that's where things are going. So how does the brain work? So that's what the brain is. How does it work? Well, okay, so Bart Simpson actually asked that question to his brilliant father. Dad, what is the mind? Is it just a system of impulses or something tangible? So what did Homer say? Relax. What is mind? No matter. What is matter? Never mind. I'm going to try to do a little better than that. So basically, the brain is an electrochemical machine. Electricity, chemicals, that actually allows the brain to work as a machine. And we know quite a bit about this process. And this really goes back to, historically, to the discovery of what was called animal electricity. So this goes back to Luigi Galvani in the late 18th century, did some really startling experiments. These experiments really, really surprised people, because what he did is he showed that he could take a dead frog, and by applying these two metals that had slightly different chemical compositions and touching them to the different parts of the frog, he could actually make the frog leg jump. And that just blew people away. In fact, it's still kind of fascinating. And he called this animal electricity. He envisioned this as being an electrical phenomenon, but not necessarily the same as this kind of electricity we think about. It had to be sort of a different kind of electricity. And I apologize for doing this, but I've showed some of you in the previous course, but I still think this is kind of a cool video I made, in which I just show how you can do that. So there's a dead frog sitting there, and what I'm doing is I'm touching it with a, oh, well, we didn't find it. Okay, so we're not going to see that video. What I do is I basically repeat the Galvani experiment by touching the frog with two electrodes that are just providing a little electric current, and the frog leg jumps. And depending on where I put that, I get the frog leg to do different things. Okay, I took a while to put that together, but so I wanted to show that to you. But it's even today, it's still kind of startling. You see that and you go, wow, that is really amazing, because the frog leg moves just like it would if it was a living frog. But we know it's not. It's a dead frog. And all we're doing is we're adding electricity and making it move in a way that's very much the way the frog would move its legs, at least, when it was still alive. So that was Galvani, and then along came several other investigators through the years, Alessandro Volta, Dubois Ramond, von Helmholtz, and others. And they actually went on and took Galvani's work and actually showed that what he called animal electricity really was just electricity. It wasn't anything unique to animals. It was the same kind of electricity that we are familiar with. Kind of ironically, Galvani never, even despite the evidence, never really gave up on his idea that what he was seeing was actually a different kind of electricity, animal electricity, sort of a vital force. In fact, what we know now is that every cell in our body actually functions like a battery. And this is not just nerve cells and muscle cells, but this is all of our cells actually have little batteries built in. And that electrical potential that is generated by these batteries actually does all kinds of things in the animal. And probably the most dramatic is that it allows signals to be passed through the nervous system as well as muscle cells. Actually, Volta created the first battery, showing that you could generate electricity by just connecting two solutions that had different concentrations of electrolytes or different concentrations of salts. And that's still how your car battery works. You basically have a cell that has a different concentration of ions in it with another cell, and that generates electricity, that generates the voltage that starts your car in the morning. So electricity is the underlying, and this idea was so captivating that, especially around the turn of the early 20th century, that idea was really sort of starting to catch on, and people were becoming really fascinated by it. And Mary Shelley's book, Frankenstein, really was a really good literary example of how people were so captured with the idea of electricity. That just seemed so weird to them that her idea is, well, if a little electricity makes a frog leg twitch, maybe a whole bunch of electricity, like a lightning bolt, will make a person completely become reanimated and become live again. Right? So what does electricity do? What is the job of electricity? Well, first of all, just a quick, let's see, there is a pointer here, right? Where did that go? Oh, here it is. Okay. All right, so this is a prototypical cell, neurons, and you can see that this part of the neuron, which is called the soma, which is actually just Latin for body. Or is that Greek? I don't know. I think it's Latin. I'm not sure. But it basically means the body of the cell. It contains the nucleus, which is where the DNA is, where all the instructions are. And really, this part of the neuron is identical to every other cell in your body. I mean, all of your cells basically have what's called a cell membrane. So it's a structure that basically separates the inside of the cell, called the cytoplasm, from the outside of the cell. And then within that is a nucleus, and that contains the instructions that the cell needs to make the appropriate proteins that allow that cell to carry out its functions. Okay, that's basically cell biology in a couple sentences. What's outside and then inside the cell is basically water with salts dissolved in it. Okay? So the inside of the cell has, it's aqueous, it's basically water, and then it has a lot of ions in it, which are, so for example, if you took table salt, sprinkled it in a glass of water and stirred it around, it would actually dissolve, right? Well, what would be in your glass of water that had after you dissolve the salt in it? Basically just be water with sodium ions, which are positive ions, and chloride ions, which are negative ions, and they would just be floating around in the water. So that's what I mean by a solution of ions. And that's really what both the inside of the cell is made up of, as well as the outside of the cell. If you've ever tasted your blood, it's salty, right? Okay. The blood is actually roughly the same composition as the fluid out here outside your cells. And it's got a lot of other things in it as well, but the primary thing is it's salt water. Okay? And similarly, the inside of the cell is salt water. Between the inside and the outside is the cell membrane, and the cell membrane is made up predominantly of fat. Okay, so fatty acids make up the composition of that cell membrane. And just like the wax, when you get your car waxed and you come out of the car wash and then it rains a little bit, what happens to the water? It beads up. Okay, so fats like wax and water don't mix. They separate. If you have vinegar and oil, they'll actually separate after time. We'd say that fats are hydrophobic, which basically just means hating water, literally hating water. They probably have that feeling of hating water, but they actually will basically separate from water. Well, that's significant because the inside and the outside of the cell are separated by waxy material. And its job is to keep these solutions separate, and particularly to keep the water and then the dissolved ions from going back and forth. So these two compartments are very separate from each other. There's the internal compartment and the outside, and they're kept different. And that cell membrane is what allows that to happen. Okay? A couple other things about the neuron. I say I have 5 minutes? Something? Okay. So a few other features of the neuron. So this part of the what's called the soma is, again, it's the same thing you find in any cell in your body. What makes neurons unique is that they have these extensions, these projections, called dendrites, and so they extend up like this. Dendrites comes from a tree, and it looks like it's tree branches, so it has that structure. That's why they're called dendrites. Most neurons also have one long projection, as shown here, that's called the axon, and that projects out and then allows the neuron to interact with other cells at the nerve terminals. These axons can be extremely long. So for example, you have some motor neurons that control, like say, muscles in your leg. And this part of the neuron, the soma, is up in your spinal cord. And the axon projects all the way down to your big toe. So you have a cell that actually can be several meters long, or not several meters, none of us are that tall, several feet long. If you're a giraffe, though, it actually is, it is even longer than that. It's several meters long. So these can be extremely long processes. Some of the longer ones have this wrapping called myelin, which actually facilitates the movement of the electrical signal, which we'll talk about in just a second. Okay? Questions about that? And I didn't specifically say that, but obviously if you have any questions, please raise your hand and I'll try to answer them. So you ready for the quiz? Okay. So most of what we know about this process of the electrical processes that happen in the brain actually were discovered on a squid. So you go, why in the world was that the case? Well, first of all, the way biology, neuroscience works is because of the evolutionary relatedness of all living things, we can learn general properties in pretty much whatever species we want. So at least at the beginning, when we're just trying to get the general gist of how something works, the way a squid does it is going to be pretty much the same way a human does it, at least in terms of the cellular events. That might seem kind of strange, but it really does work. After you figure those basic things out, then you work on the details that distinguish the different species from each other. But amazingly, we're not that much different. Some of the differences are really fairly subtle between species. But anyway, what makes the squid such a useful organism to study is because it has these giant axons. And by giant, I mean that these nerves with their axons are about 1 millimeter in diameter, which may not sound giant, but we're talking about a cell here, which normally are thousands of times smaller than that. So they just happen to have these really large axons that made them relatively easy to investigate this process of how this electrical signal works in neurons. And so people, including my postdoctoral mentor, worked on this way back in the 60s. They would go up to Woods Hole in Massachusetts, where the trawlers would go out and bring back in the squid, and then they would actually take the squid off the boat and they would actually do their experiments on the squid and really figured out most of what we know about that process, which I got 2 minutes, you think? Okay, so I'm just going to give you a quick summary of what I kind of want to do this. I'm scared this isn't going to work. All right. That was easy. All right. So here's what a nerve cell would look like. There's the soma, the cell body on the left, and then that projection, that axon that's sticking out to the right. Okay, and I've highlighted the cell membrane. That's the waxy, fatty material that separates the inside and the outside. I need to just add one more thing. So those are the ions, and I've just drawn them, symbolized them with plus and minus charges. And so these are the salts. This is the sodium and chloride, and there's some potassium and calcium. But they're basically just dissolved salt. And they basically, I colored the ones on the outside of the cell black and the ones on the inside red, just so you remember that they're not, the compositions are different. They're not exactly the same. So that's really an important feature of how cells work, is that they keep the mixture of ions different on the outside and the inside of the cell. And so here's what happens. Okay, so then the other little piece of the story that I have to tell you really quickly is that the cell membrane is a barrier. It keeps the materials moving back and forth. But what happens is that within the cell membrane, and this shouldn't have happened, but it did. Okay. So what happens is that there are these proteins that sit in the membrane of the axon. It's not letting me do that alone. And so maybe that's a good time for a break. So we'll come back to that. I can work on this technology well.
Thank you for taking your chairs. And here again is Clark Lindgren.
Is there a light here?
Yeah.
Okay. All right, so I think this is going to work. We'll see. So if the cell membrane was just this waxy material, nothing much interesting would happen. But within that membrane are proteins. And these proteins form channels. They're called ion channels. And they actually do allow ions to move across the membrane, either from inside to the outside. And they can do that in a very specific way. So they can particularly pick and say, I'm only going to let sodium go through, or only chloride, or only potassium. So that's really kind of where the cool stuff happens, because these proteins can be very, very specific and much more precise in terms of which ions they allow through. Furthermore, these protein channels have gates that can open or close. So they're not just stuck being in one particular conformation. The gate, just like a gate on your front fence, can open and it can close, and so these gates can be open and closed. And so what happens is when, if some stimulus, I'm doing this gingerly because I don't want to just, okay. So some stimulus, it triggers an event at one end of the axon, so say basically where it starts. That causes the channels to open, and it allows some of these ions to move across. Okay, I'll try to see if I touch it, that's my problem. Okay. Well, that's, hopefully you can get that good enough. So ions move through from one side to the other side, and that creates a little current. So moving ions is an electrical current, just like the current that runs through your toaster in the morning. That current actually makes a little voltage change across the membrane. And the important point about that is that triggers the next little bit of membrane to do the same thing. So that it does the same thing. So those channels open and do the same thing. They cause this little tiny current, which causes a voltage, and that causes the next, and the next, and the next. And that flipping, those little tiny currents actually move along the axon, kind of analogous to a fuse. You light a fuse and basically the spark moves along the fuse, or really a better analogy I think is dominoes. So you imagine you have a line of dominoes and you tip one, tick, 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 and then it moves all the way down. That's really how this electrical signal moves along these things like axons. Now, that has some important consequences that might not be immediately apparent. The speed with which that happens, how long do the dominoes take to fall if you're going from your spinal cord all the way down to your big toe? Well, it actually takes, it's fast, but it's not as fast as you might think. So the electricity in your radio or microphone or whatever moves really fast, about the speed of light. Basically, you think of electricity as being sort of instantaneous. You turn a switch and things just sort of happen instantaneously. These signals take a little bit of time. Anywhere from the range of the conduction velocity is anywhere from 0.5 to 100 meters per second, which really ranges from like a brisk walk. You can go about 0.5 meter per second if you're walking briskly outside, to 100 meters per second. That's a fast car. Okay, that's a race car, but it's finite speed. It's not instantaneous. It's not like what we normally think of as electricity. And that has consequences, especially for big things like ourselves, big organisms, because these signals have to pass across significant distances, and it takes some amount of time. And so that's actually a kind of an interesting problem, which we won't get into in this course, but it's a really interesting problem about how we deal with that. One simple example is, just take, like if I do this, if I hit my hand against the desk, I have the sense of that in my fingers. They feel that touch. I hear it, and I also saw it. Well, each of those three modalities are coming from different places in my body, my eyes, my ears, my fingers. They get to the brain at different times. And we still don't know how that does it, but the brain has to figure that out. And basically what it does is it kind of waits for the slowest information to get there before it makes sense of it and puts together an entire picture for us. So there's really some fascinating things about how this electricity works that aren't really immediately obvious. But that's basically how electrical signals move through nerve cells and what is responsible for brains to function. Okay, and that was learned in the squid. So that describes the electrical part of electrochemical. What does the chemical side mean? Well, that actually refers to how signals then get passed from one nerve cell to another. So here's a picture of one nerve cell up here, and you can imagine we start an electrical signal in the soma, the cell body. It travels down the axon, reaches the end nerve terminals, and then the signal jumps to the next cell, and it goes on. And that process happens at what we call the synapse. So if that, and we have we have a presynaptic cell and then a postsynaptic cell, so that terminology hopefully makes sense. So if the squid was the organism that we learned about the electrical part of the electrochemical signaling in the brain, what was the organism that was used for understanding the chemical events? And I just realized I wanted to say something else before I did that. I apologize. So first of all, we had to basically have the we had to have the Luigi moment of sort of realizing that electricity played a role in how the brain works. Well, we had an Otto Louis moment in terms of telling us that it wasn't all electricity, that there's also was a chemical element to that. And this is just such a cool experiment I have to show it to you. What Louis did is he took advantage of the fact that if you take a heart out of an animal, and this could be a human, it isn't normally done that way, but if you take the heart out of an animal, it'll keep beating. And if you're careful when you take the heart out so that you still have some of the nerves hanging on, like the vagus nerve, then you can actually stimulate the vagus nerve and it will do what it would normally do in the animal to slow the heartbeat down and also reduce the force of contraction. We actually do that lab at Grinnell. I have my students do a lab kind of like that where they study the process of controlling the heartbeat. So here what you can see is he stimulated the vagus nerve, and what's being shown right here is the contractile force, and you can see that it gets lower and then it comes back up again after you stimulate. So that's a pretty normal phenomenon. What he did that was unique is he coupled that heart to another heart, and the only thing that was connecting those two was the solution that was outside the first heart. So whatever was there in solution, when it got transferred to the second heart, it caused it to slow down as well, with a little bit of a delay. So a really, really elegant experiment, and the most straightforward interpretation is that there had to be a chemical. There was something that was being transferred from the fluid surrounding the first heart to the second heart that made it slow down. And that was basically, he called that vagus stuff, which for the vagus nerve, and now we know that the chemical is a chemical called acetylcholine. This is a significant experiment because even though people at that time, prior to Otto Louis' experiment, suspected there was this chemical basis to brain function, the electrical idea had such cachet, it was such a cool idea, that even scientists had a hard time grasping that it wasn't all electricity. We have that tendency to be polar, right? So it's either one thing or another thing. We don't really like to think about it being possibly two things, and that was actually true back then. So everybody was so persuaded that animal electricity was the thing that gave allowed brains and animals to work. They were resistant to the idea that, oh, and also there might be chemicals as well. But Louis' experiment really persuaded people. Now, the squid was the preparation that was used to figure out the electrical side of things. What was the organism used to figure out the chemical side? Frog, the northern leopard frog. I actually still use these organisms today in some of my work. They're getting harder and harder to get because of as a species that's in trouble. And many have described them as being sort of the canary in the coal mine. The frogs, they have a very permeable skin, so they're very, very sensitive to the environment. And we should be worried that we're losing our frogs because that could be a harbinger of eventually losing ourselves. Even though we have thicker skins, we're eventually going to probably suffer the same fate that frogs are suffering. But anyway, we can still get them, we can still do experiments on them. Well, why the frog? Well, it's kind of the same explanation as to why the squid was used. The frog has these synapses that are called neuromuscular junctions that connect a nerve, and so this is the nerve axon shown here in this micrograph. And it comes down, this big thing here is a muscle cell. And so you can see that the nerve axon comes down, and then these little things here are the nerve terminals. And this is huge. Like the muscle cell is a really, really big cell, so it's relatively easy to study. And the nerve is also really long. So basically for the same reason the squid was used, the frog was used. And again, for the same reason that we were able to learn a lot about the human brain in terms of its electrical properties from a squid, we actually were able to learn a lot, and still are able to learn a lot from the frog, because the way the frog does it isn't fundamentally that much different than how our brains work. Okay, so now, some fear, trepidation. Try to do this. All right. Okay, so how does a synapse work? So bear with me. All right. On the left is a typical presynaptic terminal. This could be the presynaptic terminal that we saw in that micrograph that goes from the cell bodies up in the spinal cord of the frog down to the muscle, say the leg muscle. And then the postsynaptic side is the muscle cell in this case, although it's a proxy for any postsynaptic cell. They basically work the same way. The only difference is that normal postsynaptic cells don't contract like muscle cells do. So when you study this process using a neuromuscular junction, you have to paralyze the muscle so that it doesn't do the contracting thing, because that's a little bit problematic. But all the events that precede that are identical to what happens at sort of a generic chemical synapse. So we can actually use it to learn about the process of chemical synaptic transmission. So what you notice, first of all, is that the two cells, so the pre and the postsynaptic cell, are separated. They're not physically touching each other or connected. So there is actually a gap, called synaptic gap, which is the distance between those two. So an electrical event in the presynaptic cell can't get directly transmitted to the postsynaptic cell of these chemical synapses. There's a gap between these two cells. What happens, and you'll notice that there are these little tiny balls that are here. These are called synaptic vesicles. And they are made up of, they're little balls that have the membrane that surrounds those is similar to the membrane that basically separates the inside and the outside of cells. So they actually are able to contain things inside their structures. What they contain, so little dots in here, so these synaptic vesicles contain a chemical which we call a neurotransmitter. And you've heard that term, I'm sure. So there's a neurotransmitter that's inside these synaptic vesicles. And then one other thing I'll point out is that on the other side of the gap, these structures right here, these are called neurotransmitter receptors. And you can think of the receptors as like a lock, and the neurotransmitters are like keys. And so the neurotransmitters are a specific key that will open up a particular receptor, just like a particular key will open up a lock. I use this analogy all the time in class, and I'm finding that I'm going to have to find out a different analogy, because what's the problem, right? Nobody has keys anymore. These students say, what's a key? What do you mean, my key card? So I don't know if anybody has any ideas about what I can use, but the idea is that there's a matching, and there's a matching of the structure of the key to the lock in the same way that there's a matching of the chemical neurotransmitter and the receptor.
Backing up, the items inside the cell were different than the ones outside the cell.
Yes.
And then those outside were brought into.
Yes.
How did they connect, or did they change the outside ones to inside ones, or did they create new ones?
That's a good question, and I skipped over that kind of for sake of time. But there are pumps in the membrane that restore those ionic gradients. So those ions that moved in during the electrical nerve impulse, or what we'll call the action potential, they get pumped back out again. It's more slowly, but they get pumped out so that they can restore the original environment.
Sorry, I asked.
And they're pumps in the same way, like I can think of it as like if you had a rowboat with a hole in it, and water was coming in. If you had a little pump there that kept pumping the water out as fast as it was coming in, you could still basically maintain your floating in the rowboat. And cells do the same thing. They have these pumps that are maintaining the original state. And I might have misspoke a little bit, because it's not actually completely different ions. That's part of the story, but they're also at different concentrations, and so that actually what makes them different as well. So the black versus the red plus and minuses, that was a big shortcut to not only describe the fact that there were different ions, some would have different characteristics, but they also have different amounts of concentrations, and that all gets restored later on. And that takes energy, so your brain is an extremely expensive organ. It's like maybe up to half of your energy actually gets used to drive your brain. And you look at it and you go, well, what could be taking all the energy inside this little 3-pound organ? And it's actually mostly the energy that's needed to maintain these ion gradients to what they were initially. George?
Clark, just to pursue this a little bit,
Yeah.
experimentally, how do you discover that or verify? Obviously, I don't think you see it, but is it a matter of analysis of the solutions at different points? Or?
Sure, so that was actually one of the things that the squid was good for. So because it has, thank you. So the question is, how do you experimentally figure this out? How do we know that the ions are different between the inside and the outside? And why? And basically then a corollary of that is, how do we know that makes a difference? How do we know that that different ion composition is important? And that was actually where the squid was really important, because it's such a big axon. Can't really draw it. Well, actually, if you imagine this is a squid axon, what the experimenters could do is they could dissect the axon out of the squid, put it in a dish, and then take something like a little mini rolling pin and push all of the inside cytoplasm out of the squid like you would roll, like imagine a toothpaste tube that you use properly, which means you roll it from the end. That's basically what they did, is they pushed all of the normal cytoplasm out, and then they took a syringe needle and filled it up with whatever they wanted. And so they were able to, just measure that. They could see, they could chemically measure it and figure out what was in there. And then that allowed them to manipulate these ion concentrations and ultimately reconstruct the normal sequence of events. So that's kind of a quick answer to that. And you could relatively easily change the solution outside even tiny cells, and so you can actually kind of get at that a little bit. You can find out, for example, that if you take all of the sodium out, then everything stops. So really you know that sodium ions, the main ingredient in salt, is one of the key ions. But then you can also sort of manipulate that differently.
Yeah.
Okay. So right at that point, so let's go back to the action potential, and I forget if I did use that term. Action potential just refers to that movement of the nerve impulse. And it's called an action potential because it's a potential, like an electrical potential, but it has action to it. It's moving like dominoes falling. And so I'm going to sort of draw that over here. So the action potential starts back at the cell body, and it's been moving down the axon at 40 meters per second, relatively fast, but not supersonic speed. And then it reaches the end, where it finds another set of specialized proteins, and I'm going to draw those right here. And these are called calcium channels. What that means is that these are proteins that are specialized to only let calcium ions through. So calcium, you can't even read what I wrote, can you? Shoot. So that's Ca, which stands for calcium, and 2+ means that it's an ion. And it actually has 2 positive charges, so it's a really super ion. Okay, so that's basically what my nomenclature is. These channels will only let calcium through. And what happens is when the nerve impulse reaches the end, it opens up these channels. They have gates like all these ion channels do, and they're normally sitting closed. When the action potential reaches the end, or the nerve terminal, it causes these proteins to open up their gates and they let the calcium come in. Okay? And then,
I
yes?
have trouble understanding experimentally. How do you know that the data is there? How do you experimentally? Tell me about it.
Sure. So the question is, how do you experimentally know, well, that there are proteins that are doing that, and they have the gates? I'll answer that in reverse order. I'll start today and then work back. So today, the reason we know that is because all of these things, all these proteins, we know their structure down to the specific amino acids, and even we know how they fold together. And that's because of the techniques of molecular biology, which lets you figure out what these genes are that make these proteins, and then from that, you can figure out what the amino acid building blocks are, and then from that you can make some guesses as to what it looks like structurally, but then you can actually do things like X-ray crystallography. So basically today it's a relatively straightforward thing, we just look at it. So the answer to your question is we can actually see these things. Now how do we know they have gates that open and close? Those kinds of experiments are done under situations where you can really precisely control what's happening across the synapse. And what I mean by controlling it is you can stick electrodes inside these things. And then you can not only record these electrical signals, but you can manipulate them. And so basically by manipulating these electrical signals, you can say, oh, when I do this to the cell, it lets calcium in. And you can measure that as a current, or nowadays you can measure that fluorescently. You can actually use these probes that detect calcium and light up. So your question is right on, and it's actually something that is most of us do all the time. We're always kind of doing this. So I don't make assumptions. So if I'm looking at a particular synapse and I want to know what's happening, I will either like electrically manipulate the synapse in such a way that I can basically be sure of what's happening, or I actually have this really fancy, dancy fluorescent microscope that I use that looks at a fluorescent signal, and that's because my chemist friends have created these molecules that fluoresce differently depending on what's happening in their environment. It's pretty cool. It's really good. But it's exactly, yeah, so basically these things have all been really figured out. And I don't think, going back historically, one of the fascinating things is that these, Hodgkin and Huxley were the guys who figured out the nerve impulse, and some of the other people. They didn't have a lot of these fancy molecular tools that we have today, and they actually had to make much more, they had to be much smarter than we are today, because they actually did figure out a lot of this with much less direct measurements than we have today, but they were actually able to do it by just making really careful observations and then creating models that was kind of like a hypothesis about what they think it would look like, and then you test that by manipulating it different ways and seeing if it responds the way you expect. So they're the ones who really did the incredible work in figuring all this out. We're just coming along doing the easy stuff now. Rubbish. Okay?
Thank you.
Calcium is coming from the gap then, or from the outside the cell?
Well, the gap is the out. So the question is the calcium coming from the gap? And yes, but that is also the outside of the cell.
Right.
So, and I'm glad you mentioned that, because that's an important point. So notice the synaptic gap is continuous with outside the cell, which is continuous with your bloodstream, which is continuous with what everything your bloodstream interacts with, which is like your gut and so forth. So we'll come back to this point a little bit later. This is actually one of the ways synapses will break us, is that, well, I haven't finished explaining the whole thing, but you probably can guess where this is going. Let me do that, and then I'll come back to that point. So the calcium comes in, and the calcium causes these vesicles to fuse with the membrane and then release these neurotransmitter molecules into the gap. Okay, so they just get dumped into this gap between the two synapses. They diffuse around, they basically just diffuse from one side to the other, but they end up hitting these receptors. And that's where the lock and the key works. So then that opens these receptors, and lo and behold, these receptors are ion channels. And so when they bind these neurotransmitters, they open up their gates. It's all gates. They open up their gates, and then they allow ions to move in. And that triggers an electrical change in the postsynaptic cell, which then leads to activation, or leads to an action potential in the postsynaptic cell. So the sequence goes like this. Electrical event, action potential in the presynaptic cell opens up these calcium channels. These vesicles fuse and release their contents into the gap. The neurotransmitters then diffuse across just by this random diffusion. Things will diffuse from an area of higher concentration to a lower concentration. Just like if I opened a bottle of perfume up here, I would smell it right away, and then maybe eventually, it's kind of a neat demonstration, I should have thought of doing that, but if you open up a thing of perfume and then I have you raise your hands when you smell it, you basically go back to the room until eventually you smell it. Well, what's happening is the perfume becomes volatile, comes into the air, it's a high concentration here, and then it basically diffuses from high to low. And so basically, eventually it would reach you at the back row because you're farthest back and it's taking the longest time to diffuse. Now, it might not get all the way to the back row if you didn't have a lot of perfume, but that's basically how diffusion works. And it's the same thing here. So the nerve terminal releases a whole bunch of these chemicals, and then they just diffuse. And these receptors are located right across the gap. So they're not terribly far away. They're probably more like the second row here. So they diffuse not very far away, and they basically bind to these receptors, and then these receptors are their own kind of ion channels that open up, and then they trigger action potentials to occur in the other cell.
So, I mean, in a simple, for my simple mind, that everything within the cell is electric, the connection is chemical.
So everything, I'll answer the question, but restate, I mean, this is, so the idea is everything that happens intracell or within the cell is electrical. And then information transfer between cells is chemical, yes.
Yep.
And there are some exceptions to that. But there actually are electrical synapses. They're not as many of them, but there actually are cells that actually are connected up electrically as well. But they're relatively less. Okay. Couple more things here. So let's see. So we have an action potential opens up the calcium channels. They allow the neurotransmitter to diffuse across to the receptors, binds to the receptors. And then the receptors then basically trigger another electrical event in those postsynaptic cells, which then carry on to the next, to the next synapse. So then that electrical event in the postsynaptic cell then goes on to the next cell in the sequence. That process gets repeated again. So that's where these networks come along. And all of these things are happening. So your 10 to the 15th synapses are doing this all the time. By the way, Earl pointed out that the number of stars in the Milky Way is only 10 to the 11th. So we got like 1,000 more connections. I think I said there's a few more. There's a few. All right, so, and that event is happening over. That's happening. I mean, you can't get your head around that. It's happening all the time in all of your synapses.
Does this diffusion affect the glial cells, or?
Oh yeah.
You're not going to talk about
so does this diffusion affect the glial cells? And then we'll be here forever if I try to explain that. but it does. And so to quickly answer that, this is referring back to an earlier bucket course I taught, where I actually talk about glial cells. But they're actually sitting around, and they have receptors for these neurotransmitters as well. And they also basically get activated. But one thing that's a little bit different is that those receptors in the glial cells don't necessarily open up ion channels. They trigger other events that are more chemical in nature inside the glial cells. So it's a little different story, and I'd rather not. I'd like to go there, but I won't do it right now. Okay. Now, one thing that you might think there's something missing here. So I just described for you a signaling process, right? I described to you how a signal goes from one cell to the other. What happened I showed you that's critical for this thing to work as a signal?
Stimulus.
Let's see. So we know how to turn on the signal. What do we need?
Turn it off.
We need to turn it off, right? So if you're going to have a signal, like think of a light signal that you're using to flash Morse code to somebody across the town, you need to basically turn it on and then turn it off. I mean, it doesn't make any sense. You can't have a signal that just stays on all the time. It's only going to work once, and that's not going to be very good. So you have to basically turn the signal off. So how do the synapses turn off? It turns out there are 3 different processes that turn the signal off. One process is just simply the neurotransmitter molecules diffuse away because, again, open up the perfume, it would eventually diffuse out to the little library, and there wouldn't be very much left anymore at the front row or second row, and so basically that would be one way of stopping the process. So these neurotransmitter molecules can just diffuse away. They can also be taken up, they can be retaken back up into the nerve terminal. So this is called a reuptake process. So these neurotransmitters can be pumped back, like recycled, if you will. Our synapses are the great recyclers, and so they basically will take these neurotransmitters and recycle them back and then put them back in the vesicles and use them over and over and over again, so we don't waste our neurotransmitters. And then the third method is to chemically modify the neurotransmitters so they don't work anymore. So basically break them down so that they are chemically inactive. Turns out that the neuromuscular junction uses all 3 of those mechanisms. Some synapses will only use diffusion, some will use reuptake. At the neuromuscular junction of the frog as well as the human, what happens is this neurotransmitter, which the name of it is called acetylcholine, which I'll just abbreviate ACH, which stands for acetyl, which is actually acetate. So if you think that's that's a base, that's like the acid that's on in vinegar, for example. So acetic acid is basically one part of this neurotransmitter molecule, and the other part is choline, which is a little more complicated structure. And what happens is that there's an enzyme that's produced by the muscle cell, and it's secreted out into the synaptic gap. And this enzyme is called acetylcholinesterase, which I'm abbreviating with a little e. And what it does is it takes the vinegar part, it takes the acetic acid and clips it off of the choline. So it breaks acetylcholine down into acetate and choline. And then the choline is what actually gets transported back into the nerve terminal. So it doesn't actually take back fully formed acetylcholine, it just takes back the choline. Acetate is a really pretty ubiquitous chemical inside cells. It gets its acetate from somewhere else, puts it back together again, and sticks it in the vesicle for future methods. Now, why would the neuromuscular junction be so obsessed with turning off? So I mentioned all synapses have some form of inactivating the neurotransmitter, because otherwise it wouldn't be a very good signal. Neuromuscular junction goes kind of crazy, right? They just, it's like, all right, let's first destroy the neurotransmitter as quick as we can. And notice where this enzyme is. Does that seem a little bit strange to you? This inactivating enzyme is sitting between the pre and the postsynaptic elements. So that means that the neurotransmitter that gets released has to make it past this degradatory enzyme before it even can bind to the neurotransmitter. I have students do this little activity where they actually, I dress them up in outfits that reflect the different parts of these, that some of them are neurotransmitters, some of them are receptors, others are these inactivators. And I line the inactivators. I usually try to get like the football players to be the inactivators, so they look kind of intimidating there. And the neurotransmitters have to get past them, and there's rules that prevent that from being a massacre. But it illustrates a really important point. Half of the acetylcholine that you release, every time you move a muscle, half of the acetylcholine you release gets destroyed before it ever binds to a receptor, because half of them get, and I think of an army rushing, to conquer a fort. Imagine half of the soldiers get killed and only half of them make it to basically, scale the walls or whatever they're going to do. It's astounding that we're, you might say, wasting so much. I mean, nature's pretty thrifty. It's been, weaned over the years to basically not waste any, because it costs energy for cells to remake this acetylcholine and other. So why would we be so wasteful? Well, it must be pretty important to turn the synapse off, right? Because we're not even going to wait. Was there a question that I didn't answer? Oh, it's the wrong one. So why would we do that? Why would we be so wasteful? I like to use the analogy of, let's say, Grinnell College, the president's putting together the budgets for different departments and decides what biology needs $100,000, but we're just going to give them $200,000 because they just waste half of it anyway. But probably wouldn't happen very, very, very likely. So we are designed to really turn off that neuromuscular junction pretty heavily. And so we not only have the mechanisms that are found in all synapses, but this one in particular, we turn it off. And can you think about what muscles do? Like, why would that maybe be pretty critical for how muscles would work?
Relax it, yeah.
Right, because you excite your muscle, but then it has to relax. And basically you can't, every time you move a joint, you're activating muscles on one side of it and you're relaxing the ones over here. So relaxation is as critical as contracting, really, for movement. And imagine playing the piano, doing all kinds of things that require pretty sophisticated, fast motions. You've got to turn the signal off really, really fast. And so chemical signals are slower. I think somebody mentioned that. So electrical signals are fast, and they turn off really, really quickly. But chemical signals, in order to make those fast enough to accomplish certain things that these synapses have to do, there's special machinery built in to basically make that happen. Yes?
When they don't turn off, is that what causes a Charlie horse?
If they don't turn off, does that cause a Charlie horse? I'm not sure. I actually have to think about that, but we'll come to that. There is a really key consequence of them not turning off. I'm going to save that for later. Yes.
In heart treatments, they give you something called calcium channel blockers.
Yeah.
What do they block with?
So they block with calcium channel blockers. So the question is, I'm sorry. So in people who are getting, who have different cardiac difficulties or challenges, a lot of times you might just get regular calcium channel blockers. And what those are doing is they are blocking calcium channels in heart and in vascular tissue as well.
Well, what do they block with?
The chemical, so what happens is the chemical that's used physically binds to the protein that makes up that channel. And then when they're bound to it, they prevent the channel from letting calcium go through. There's a couple different ways that they work. One way is they just sit in the way of the channel. They just basically physically block the channel. So even if it opens up, the calcium can't come in because it's basically being physically prevented. And other ones will basically muck with the gate or something like that. But those are different kinds of calcium channels, but really similar. They're very similar to the ones that I'm talking about. Those are just ones that are in the heart. And heart muscle, so the other part of the story, which we didn't get into, is that contraction of muscle requires calcium as well. So calcium comes in a couple places. One, it comes in activating the muscle in the first place, but then it also comes in and being necessary for the muscle to contract. And that's the part that's actually being blocked by that. But this whole beautiful, wonderful world of pharmacology allows us to be much more specific. And so you can take a calcium channel blocker that blocks the types of proteins that are in your heart, and those don't block the proteins that are in your nerve terminals. Am I doing that? I'm almost ready. I'm almost at a good stopping point. So, any more questions about this, though? These are all great, excellent questions.
How does the time for transmission down the neuron electrical signal compare to the time to go across?
Oh, that's a good question. So how does the time compare for the time of the electrical signal to go down to the time it takes the chemical signal to go across the synapse? And the chemical signal is quite a bit slower. So for example, with the neuromuscular junction, 2 milliseconds is the amount of time it takes to go from the action potential reaching the nerve terminal to getting to the muscle. Whereas it's microseconds to basically move down the axon. And in fact, experimentally, we use that feature because if you have a circuit and you want to know, you want to make an approximation as to how many synapses go between input and output, so you have like the knee-jerk reflex where you tap on your patellar tendon and then your knee jerks out like that. You can actually measure that delay, and because the action potential is really super fast, you can actually count up how many synapses are there based on how much the delay is. Roughly 2 milliseconds per synapse. And what we find out is that that knee-jerk reflex only has 1 synapse, which is actually one of the ways you can turn it on. Am I done? I'm hitting the hook. Okay, so we're done. So
I tell you, I was so fascinated with this that I forgot to watch the time, so we're lucky this is 11:45. Thank you so much, Clark. We'll see you all back here again next week. And in the meantime, you may want to go to the website and see the video of our class this morning for a refresher course, because you never know if there's going to be a quiz. And the cards that you have on your chairs this morning give you the website, so you can go there, and I think probably by tomorrow, Bob, we will have the video up. Thank you for coming this morning. We'll see you next week at the same time as today. And thank you, Bob.